Chapter 27, Protein Metabolism. Protein synthesis is one of the most complex biosynthetic processes in our body. Despite this complexity, proteins are made at exceedingly high rates. To maintain the appropriate concentration of proteins, our body must keep up with protein synthesis as well as protein degradation rates. In this chapter, we will discuss three important aspects. First, how proteins are synthesized, how proteins are targeted, and how proteins are degraded in the cells. In part 1 of this chapter, we will discuss the first step, how are proteins synthesized, and what is the basics of translational machinery. To start with, we'll discuss genetic code. The learning goals of this chapter are as follows. First, we will start by discussing the genetic code. By the end of this chapter, you will understand as to how genetic code is used for protein translation. We will continue our discussion in subsequent parts to understand the synthesis of amino acyl tRNA, which is an integral part of the ribosome. And we'll see in brief as to how ribosomes function to synthesize proteins. Finally, we will discuss protein targeting to various organelles and finish off our discussion with protein degradation. Protein synthesis is highly energy demanding, and so it is highly coordinated. Interestingly, protein synthesis can use 90% of the chemical energy of a cell. Number of copies of proteins produced correspond to number of copies needed. If you consider this simple diagram, it shows a cycle, and this represents a protein regulation. When proteins are translated by the ribosomes, they are then folded and targeted to various cellular locations. And here, proteins can be modified, which is called as post-translational modification, and the function of the protein is then utilized by the cell. And once the proteins are utilized and the function of these proteins are performed, they are then degraded. And degradation has to keep pace with synthesis, and this is important for protein synthesis and the regulation of protein homeostasis. I said in the beginning that protein synthesis is a very complex process. And why is this? If you consider eukaryotes, it requires greater than 300 biomolecules to synthesize a protein. And in this case, greater than 70 ribosomal proteins are required. About 20 amino acid activation enzymes, about 20 protein factors for initiation, elongation, and termination of peptides, about 100 additional enzymes for final processing, and last but not least, 40 different kinds of transfer RNA and ribosomal RNAs are required for this process. So protein synthesis is extremely complicated. Three early advances in biochemical research has led to the understanding of protein synthesis. First, the discovery that proteins are synthesized at ribosomes. Second, amino acids are activated for synthesis by attachment to tRNA via amino acyl tRNA synthetases. And finally, tRNA acts as an adapter to translate mRNA into protein. Now, because of these three early advances, we now know that the amino acid is covalently bound at the 3' end of a tRNA molecule. And a specific nucleotide triplet in tRNA interacts with a particular triplet in the codon of mRNA. And this interaction happens via hydrogen bonding. This has led to the discovery of many different processes. The genetic code for proteins consists of triplets of nucleotides. Why? Why would you need 3 nucleotides to represent one amino acid? Here's the answer. There are 20 common genetically encoded amino acids. The 4 letters of DNA in groups of 2 is insufficient, which means it can only yield 16 different combinations. The 4 letters of DNA in group of 3, however, is sufficient, which means there are about 64 different combinations that can result. And we have only 20 amino acids. So living organisms use non-overlapping mRNA code with no punctuations. And in this case, a code of 3 DNA letters, which is called as a codon. The genetic code is written in the 5' to 3' direction. A codon, as shown here, is a triplet of nucleotides that codes for a specific amino acid. When translation occurs, it occurs in such a way that these nucleotide triplets, this is a 1 triplet, 2 triplet, 3 triplet, and each triplets are codons. These nucleotide triplets are read in a successive, non-overlapping fashion during translation. A specific first codon in the sequence establishes the reading frame. This means that this first sequence, let's assume that this is the first codon in the sequence, and this establishes what is called as a reading frame. If the reading frame is thrown off by a base or 2, all subsequent codons are out of order. For example, if the first 3 nucleotides do not form a codon, and the second, third, and the fourth forms a codon like this in the reading frame 2, then every amino acid that was originally coded by these codons would change. The amino acid sequence of a protein is defined by a linear sequence of contiguous triplets, and these triplets are the codons. So in principle, any single-stranded DNA or mRNA sequence has 3 possible reading frames: reading frame 1, 2, and 3. But only one is likely to encode a given protein. The genetic code can either be overlapping or non-overlapping. In a non-overlapping code, codons, as shown here, and are numbered consecutively, do not share nucleotides. In an overlapping code, some nucleotides in the mRNA are shared by different codons. In a triplet code with maximum overlap, many nucleotides, such as a third nucleotide from the left, are shared by 3 codons. So if you take this particular nucleotide, it can be part of 1, 2, 3, the first codon in the first reading frame, UAC, which is the second, a codon in the second reading frame, or ACG in the third, a codon in the third reading frame. In an overlapping code, the triplet sequence of the first codon limits the possible sequences for the second codon. A non-overlapping code provides much more flexibility in the triplet sequence of neighboring codons, and therefore in the possible amino acid sequences designated by that code. So to make things simple, a non-overlapping code is much more useful, because if one knows the code, it is easy to predict the protein sequence. Cracking the genetic code was never easy. One of the key questions remained was, what were the three-letter code words for each amino acids? So in 1961, Marshall Nierenberg and Heinrich Mattei reported their first breakthrough. They incubated synthetic polyuridylate, or poly-U as it is called, with an E. coli extract, a bacterial extract. And they also added GTP, ATP, and a mixture of 20 amino acids in 20 different tubes. So this was performed in 20 different tubes, each containing a different radioactively labeled amino acid. Now, because poly-U mRNA is made up of many successive uridylate, as you can see, the UUU triplet should promote the synthesis of a polypeptide containing only the amino acid encoded by the U codon. A radioactive polypeptide was indeed formed in one of those 20 tubes. So the experiment was indeed a success. And they concluded, and the tube containing radioactive phenylalanine resulted in a polypeptide. So polyphenylalanine polypeptide was formed, and only a polypeptide in that particular tube was found. So Nierenberg and Mattei concluded that the triplet codon, U, encodes phenylalanine. A complementary approach to cracking the genetic code was provided by Gobind Khurana. He developed chemical methods to synthesize polyribonucleotides with defined repeating sequences of 2 or 4 bases. And in this case, this strategy resulted in mRNAs in planned patterns. The copolymer ACN, for example, has alternating ACA and CAC codons. As you can see, ACA, CAC, it's alternating. Now, the polypeptide synthesized on this messenger contained equal amounts of threonine and histidine, and that was established from Khurana experiment. It was later found out that the histidine codon has 1 A and 2 Cs, and hence the codon CAC codes for histidine and ACA codes for threonine. Consolidation of the results from many experiments permitted the assignment of codons to individual amino acids, and that resulted in the nucleotide code dictionary, as shown here. 61 out of 64 codons code for amino acids. Now, if you count this table, you will see that there are 64 codons, of which 61 code for amino acids. Only methionine and tryptophan have a single codon. Now, methionine, as you can see, AUG, only AUG codes for methionine. Tryptophan, UGG codes for tryptophan. And so both these amino acids only have one codons. AUG is the initiation codon, as shown here. So the first amino acid that exists in a protein is almost always a methionine. And that is because AUG is the initiation codon, and the ribosome recognizes AUG as the start codon. 3 codons, UAA, UGA and UAG are called as termination codons or stop codons. An example of termination codons in random sequences is given in these three reading frames. In reading frame 1, as you can see, after 3 codons, there is a codon UAA, which is a stop codon or a termination codon. And if you get to the reading frame 2, the codon starts with a stop codon. Depending on how a ribosome reads through this, it can make a tripeptide from the first reading frame before it encounters a stop codon. In a random sequence like this, 1 in every 20 codons in each reading frame is, on average, a termination codon. In general, a reading frame without a termination codon among 50 or more consecutive codons is called as an open reading frame. Long open reading frames usually correspond to genes that encode proteins. One of the striking features of the genetic code is that an amino acid may be specified by more than one codon. The code, hence, is described as degenerate. For example, if you look at leucine, 1, 2, 3, 4, 5, 6. Leucine can be coded by 6 codons. Now, this does not suggest that the code is flawed. Although an amino acid may have 2 or more codons, each codon specifies only one amino acid. So, for example, if CUU is coding for leucine, CUU cannot code for any other amino acid. The degeneracy of the code is not uniform. If you look at methionine and tryptophan, as I said before, they have each one codon. But if you look at other amino acids like arginine, serine, and leucine that I just described, they have 6 codons. The table here shows as to how many codons are available for each of the amino acids. But remember, as I said before, each codon only specifies one amino acid, and hence the power of codons. The genetic code is universal, but it has some exceptions. It is used by prokaryotes and eukaryotes across species. Mitochondria contains DNA and use a slightly different code. UGA, which is usually a stop codon, encodes tryptophan in vertebrate mitochondria instead of stop. AGA or AGG, which encodes stop in vertebrate mitochondria instead of arginine. So these are in mitochondria of vertebrates. These are the exceptions. Mitochondria encodes their own tRNA using 22 instead of 32. tRNA or transfer RNA uses anticodons to recognize the codons in mRNA. The codon sequence in mRNA is complementary to the anticodon sequence. More importantly, the codon pairs with anticodon via hydrogen bonds. The alignment of the 2 RNA segments is antiparallel, which means that if the mRNA goes from 5' to 3' and we read the codon from 5' to 3', the anticodon is read from 3' to 5', as you can see here. The anticodon in tRNA is from 3' to 5'. We always read codons from 5' to 3'. So when we talk about anticodons, the anticodon is always from 3' to 5'. When several different codons specify one amino acid, The difference between them usually lies at the third base position. The third base of a codon can form non-canonical base pairs with its complement or the anticodon in the tRNA. Let's look at an example. The codon CGA pairs with the anticodon ICG, where I is a non-canonical base, and this base pair is a non-canonical base pair. As you can see, for all these three different codon-anticodon interaction, the first base of the anticodon is a non-canonical base. And some tRNAs contain inosinate as a non-canonical base, which can hydrogen bond with uracil, cytosine, or adenine. If you consider the structure of inosine, it's very similar to guanine, except that the NH2 is missing, and hence can form two hydrogen bonds with this oxygen and with this NH. As a result, the hydrogen bonds are weaker and were named by Crick as Wobble base pairs. Example, in yeast, CGA, GCU, and CGC all bind to the tRNA for arginine, which has the codon GCI, or which has the anticodon GCI. So the inosine in there can hydrogen bond with adenine, uracil, or cytosine. However, you may see that inosine cannot hydrogen bond with guanine. The first Wobble base hypothesis states that the first two bases of an mRNA codon always form strong Watson-Crick base pairs with the corresponding bases of the tRNA anticodon and confer most of the coding specificity. The first two bases of the mRNA codon are U and A in this case. And the first hypothesis states that these two bases form strong Watson-Crick hydrogen bonding with the last two bases of the anticodon. Now, if you look at the examples, you can see that X prime, Y prime, and XY happens to be the strongest hydrogen bonding that forms in a codon-anticodon interaction. The Wobble hypothesis 2. This has four parts to it. And the first part states that the first base of the anticodon reading in the 5' to 3' direction determines the number of codons recognized by the tRNA. When the first base of the anticodon is C or A, as shown here, base pairing is specific and only one codon is recognized by the tRNA. So if it is a C or A in the first base of an anticodon, it specifically recognizes its complementary base, A recognizing U or C recognizing G. When the first base is U or G, if it's U or G, binding is less specific and two different codons may be read, which means that U can hydrogen bond with A or G, and G can hydrogen bond either with C or U. And it also says that the binding is less specific. When inosine is the first nucleotide of an anticodon, three different codons can be recognized. So I can hydrogen bond with A, U, and C, except G. So an easy way to remember is this. Inosine looks like guanine. So inosine cannot hydrogen bond with guanine, but it can hydrogen bond with adenine, cytosine, and uracil. And if inosine is the first base of the anticodon, then it can recognize three different bases on the codon. All these 4 points form the second Wobble hypothesis. The Wobble hypothesis 3 states that when an amino acid is specified by several different codons, the codon that differ in either of the first two bases require different tRNA. For example, arginine, AGA, AGG, CGG, CGA, CGC, and CGU. There are 6 codons, 6 different codons that corresponds or codes for arginine. But if you look at these 4, the first 2 bases are the same, whereas for these 2 codons for arginine, the first 2 bases are different from that of the 4 codons that starts with CG. So for these 2, you need 1 separate tRNA, and for these 4, you need another separate tRNA. That's what this hypothesis says. If you take another example of leucine, and if you consider leucine, CUU, CUC, CUA, CUG, all four of these codons would be recognized by 1 leucine tRNA synthetase, whereas UUA and UUG would require another tRNA. A minimum of 32 tRNAs are required to translate all 61 codons. 31 to encode for the amino acid and 1 for initiation, that is AUG for methionine. This is the 4th Wobble hypothesis. I would recommend you understand these hypotheses and at least remember the 2nd Wobble hypothesis as to what inosine can recognize and how the wobbling matters, in the sense that the hydrogen bonding interactions of the first base of the anticodon dictates how many codons it can recognize. The genetic code plays an interesting role in safeguarding the genomic integrity of every living organism. Evolution did not produce a code in which codon assignments appear at random. Instead, the code is strikingly resistant to the deleterious effects of the most common kinds of mutations. The most common kinds of mutations are called missense mutations. In this case, single new base pair replaces another. However, the genetic code is resistant to missense mutations. A common one is a transition mutation, in which a purine is replaced by a purine. The degenerate code also allows certain mutations to still code for the same amino acid. In case of silent mutations, where different nucleotide in DNA but same amino acid in protein can happen. Mutation in the first base of a codon usually produces a conservative substitution. For example, substitution in the first base of the codon from GUU to AUU translates a leucine instead of a valine. Now, a valine is an amino acid that is similar to leucine. So there is not much damage that is done because the amino acid property is conserved. Both valine and leucine are hydrophobic amino acids. And so these are different ways in which the genetic code is sort of protected from mutations. Some messenger RNAs are edited before protein synthesis. Editing involves alteration, addition, or deletion of nucleotides in the RNA in a manner that affects the meaning of the transcript when it is translated. Addition or deletion of nucleotides has been most commonly observed in RNAs originating from the mitochondrial and chloroplast genomes. Editing uses guide RNAs or gRNAs that temporarily hybridize with the messenger RNA and acts as templates for editing. A post-transcriptional editing process is shown here. in the post-transcriptional editing process, 4 uracil residues are inserted into the edited mRNA, as shown here. Now, the base pairing between GU, as shown in this interaction between the mRNA and guide RNA, is pretty common in RNA. And using this guide RNA, an enzyme inserts 4 uracil into this new mRNA that gets transcribed. And this is an example of an editing process where nucleotides are added. RNA editing by alteration of nucleotides most commonly involves the enzymatic deamination of adenosine or cytidine residues, as shown here. Deamination of adenosine results in inosine, whereas deamination of cytidine results in uridine. The first step, adenosine to inosine, is catalyzed by the enzyme ADAR, or adenosine deaminases that act on RNA. Cytidine to uridine deamination is catalyzed by the enzyme APOBECS, or ApoB mRNA editing catalytic peptide family of enzymes, which includes activation-induced deaminase enzymes. Now, both ADAR and APOBECS groups of deaminase enzymes have a homologous zinc coordinating catalytic domains. A characteristics of ADAR is that ADAR converts adenosine to inosine, or A to I editing. And this A to I editing of RNA transcripts is common in primates. And most of the editing occurs in signs or short interspersed elements in transposons. And eukaryotic transposons that are particularly common in mammalian genomes get RNA edited. APOBECS, on the other hand, has various different classes, and most of the APOBECS enzymes are DNA deaminases. That means that they act on DNAs rather than RNA. Only certain APOBECS act on mRNA. An example of mRNA editing by the APOBECS enzyme is shown here. In this case, the RNA editing of the transcript of the gene for the ApoB component of LDL is shown, and one form of apolipoprotein, or ApoB100, is synthesized in the liver. A second form, called as ApoB48, is synthesized in human intestine. Both are encoded by an mRNA produced from the gene for ApoB100. An ApoBECS cytidine deaminase found only in the intestine binds to this mRNA at the codon for the amino acid that is coded by CAA, or it codes for glutamine in this case, and converts the cytidine to uracil. As a result, a stop codon emerges. So ApoB48 that is produced in the intestine from this modified mRNA is a truncated protein form of ApoB100. Now, this reaction permits tissue-specific synthesis of 2 different forms of proteins from one gene, and that is the utility of mRNA editing.